Hey guys, so I'm going to walk you through today what we're doing about this enzyme lab. It's in your packet on page 6. So we were simulating a chemical reaction by using our hands as enzymes. So one group member in the class was using their hand as an enzyme. The active site is the front of the hand where we're actually able to do a reaction. And the reaction was we were picking pennies up off the floor one at a time and putting them on the desk heads up. You had 10 seconds at a time to get as many as you could. And our first baseline, our control group, was just using our hand. Then we denatured it. So we misshaped our hand in our active site by taping our thumb down so you couldn't use your thumb mostly. Then we had a coenzyme for round 3 where you had 2 people, each with one hand, that were able to work together and be sort of a catalyst and speed it all up. And then the last round we had a competitive inhibitor. So you're holding a tennis ball in the palm of your hand, which then really limits what you're able to do because your active site is taken up. So we're not going to worry about the hypothesis for this one, but I want to walk you through if you are remote or you were not here, please make sure you're listening to these parts. So you're going to write down group data information. In Classroom is an enzyme lab data spreadsheet that's posted. You all have access to edit it. Please be careful and do not add or delete any information that you are not intending to, because everybody's gonna be using this. So I'm using third hour as an example because we just completed the lab in there. And what you're gonna do is choose a group if you are not here today, and you are gonna be writing down all of this information. So on your lab, where it says your baseline, you'll write down 6, 12, 18, 25, and go down that way for that first section. You'll notice at the end that I just continued their last number through the end of the time frame. And then your second row is denatured, so you're going to write down what group 1 did with their denatured hand enzyme. Then we have our coenzyme and our competitive inhibitor. You'll also notice that on the sides over here, these are all averages for what the class was able to do. So averaging out, in this case, the 4 enzymes that we had going at once. First hour, you'll need to wait until you have all 5 groups information in there. And then 8th hour, you will have 4 groups as well. I believe we haven't gotten there yet, but we'll get there today. And those pieces of information on those averages are going to go down in this table for class averages. So you'll notice here we have the same 4 trials and then the same amount of time frames. And you're just going to go ahead and write the class average that's calculated in the spreadsheet for you. The next thing they want you to do is to create a line graph on graph paper that shows the class average data. So you can either do this on the graph paper or watch the video that I will post separately about how to create a line graph in Google Sheets, and you can submit that as well. You will need to still make sure that you include a title, labeled X and Y axis with your units, and a color-coded key. So each of our trials should be a different color so that we can track over time what is happening with each of them. And on your x-axis, you will have the time intervals. So along the bottom here, you'll have your time from 0 to 10, 10 to 20, 20 to 30, and so forth. And then up the side, you'll have the number of pennies. So you will notice that each class is going to have a slightly different number for our total at the bottom. Hold on, something went very wrong here. It's probably 43, supposed to go right there. Whatever ending number we have down here as our total is the highest number that we need to have for our y-axis and our number of pennies, which represents our chemical reactions. So you're going to be putting dots in one color for each of the trials, not each of the time intervals, but each of the trials. So your control group, your denatured enzymes, your coenzyme, and your competitive inhibitor are all going to be different colors, and you're going to connect the dots as you go. And then you need to answer these analysis questions. I will post these in a Google Doc format on the classroom so you can type them and answer them there instead of handwriting and submitting. It helps me grade it just a little bit faster. If you do your graph by hand, then you'll need to take a picture of the graph and submit it to that assignment as well, or you can submit whatever Google Sheet file you use to create the graph. Make sure you are answering each of these parts of the questions fully, and make sure that you are relating it back to enzymes and what we're learning about. That's one problem that I'm seeing a lot with labs is we're not relating the material to what we're actually discussing in class. And that's kind of the whole point. So if you have questions, please let me know. But this should get you through completing that lab. It's going to be due Tuesday at midnight. You will have other things that are happening on Monday. So please make sure that you use your time wisely. Thanks.